Today, I will be presenting the results from the scientific article titled Meditation and Vacation Effects Have an Impact on Disease-Associated Molecular Phenotypes. The bioinformatics method used in this study was Bayesian networks, and the audience of this presentation is my BIMM143 peers. My name is Madison Baxter, and let's get started. So before we get started, here's an overview of the sections of the study that the presentation will cover. I will start with an introduction to the research, which will include the scientific question and the hypothesis. Next, I will discuss some of the key details, including the key terms and take-home findings. Third, I will discuss the importance of this research, which includes a discussion of similar research that has been conducted before and how this study improves upon this previous research, but does include some limitations of its own. I will then discuss the bioinformatics method used, the Bayesian network, and its applicability to the study. Finally, I'll provide an example of the code used in this method and dive further into describing it. The scientific question of the study is, can the effects of meditation on health be quantified by change in molecular phenotypes in relation to the effects from relaxation alone? And the hypothesis is, the effects of intensive meditation should downregulate molecular stress pathways more than the effects of relaxation alone. Before going into the details of the study, it will be important to first go over some key terms. First is transcriptome-wide expression, which is a method used in biological research where genomic information can be converted into quantifiable units that map to genes and gene expression. This is how the genetic data is collected in this study to provide quantifiable results. The next key term is blood-based biomarkers, which are characteristics of the body that are measurable by taking a blood sample. This is how the genetic data is collected in the study, both before meditation treatment as a baseline and again after for comparison. A couple examples of the biomarkers used here are telomere length, inflammation, and amyloid beta proteins. The third key term is telomeres, which are important because, as I just mentioned, one of the biomarkers studied was telomere length. Telomeres are regions of repeat nucleotide sequences on the ends of chromosomes. Telomere length is important to this study because short telomeres are correlated with disease-associated phenotypes, whereas long telomeres are an indication of better health. Next is gene expression, which is the process by which gene sequences are transcribed into functional gene products, such as proteins or functional RNAs, which in turn produce a certain phenotype. This is ultimately what the study is measuring, a change in gene expression from before to after meditation. Finally, phenotype is the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. So in this study, a change in gene expression will lead to a change in phenotype expression. For example, a change in telomere length. There are multiple take-home findings of this article. First, the week-long vacation for all three groups resulted in improved regulation of stress response and overall health. The changes in gene expression were consistent with suppression of gene networks regulating stress response, inflammation, and wound healing. Or in other words, since participants were in a less stressful state, the genes for dealing with stress, injury, and healing were downregulated. Second, the novice meditating group showed better additional positive gene expression changes than the vacation-only group, including regulation of protein synthesis and trafficking, and viral expression and viral infection. Additionally, in this group, there was also increase in telomerase activity, which means increased telomere length, which is associated again with decrease in stress-related cell aging. And third, the Bayesian network displayed these specific positive changes in gene expression for the vacation baseline and showed additional positive changes for the meditation group, including viral gene expression and regulation of telomerase. Why is this study important? Let's first look at a couple examples of previous studies conducted. The first study I will discuss involves breast cancer patients, where one group of breast cancer patients underwent long-term intensive meditation and another group did not. The results from the group that underwent the long-term meditation suggested that long-term intensive meditation could slow cellular aging rates by increasing telomerase activity and lengthening of telomeres. Both increasing of telomerase and lengthening of the telomeres are associated with lower chronic stress and slower cellular aging rates. However, while the study was long-term, it could not be conducted in a controlled environment. The second study I will discuss compared short-term meditation in novice versus experienced meditators after just one meditation session. Both groups showed positive changes in inflammation, energy metabolism, mitochondrial function, and telomerase maintenance, However, while the study could be conducted in a controlled environment, it was a short-term study since it only involved one meditation session. So the follow-up question to these studies is, how can the effects of long-term meditation be studied in a controlled environment? Well, the importance of this study is that it does just that. This is the first study to measure the effects of long-term meditation intervention using a controlled environment. This was made possible by the experimental design by using the vacation effect as a baseline for all groups, as opposed to an uncontrolled environment that is subject to outside stressors and other variables. In this study, there are 3 study groups, all subject to a week-long vacation at a resort where outside stressors become controlled, and the baseline for all groups is this uniform vacation. One group is part of a non-meditation vacation. A second group is part of a meditation vacation comprised of novice meditators. And the third group is part of a meditation vacation comprised of experienced meditators. This way, long-term effects of meditation can be studied in a controlled way. However, this study still has limitations. There is a limited sample size of people participating in the study, and all these people are female, 40 to 60 years old, and from the same geographic area. While this is helpful to compare the 3 study groups against each other, the results are not comprehensive for a wider population. Now, to introduce the bioinformatics method used, the Bayesian network. The Bayesian network is a probability model that connects different variables called nodes by links represented as lines or arrows. These links provide insight to how certain variables affect others using probability. Take a look at this simple example of a Bayesian network on the right. Here you can see 4 nodes, A, B, C, and D. These all represent different variables. They are connected by links, which are the arrows. A is an independent variable affecting D and B, which both affect variable C. Here is figure 3 from the study, which shows the Bayesian networks created for the study. There are 2 different networks created here, one for the non-meditating group on the left and one for the meditating group on the right. Different variables, in this case, groups of interconnected genes associated with biological processes, including molecular functions and cell cycle pathways, are shown in individual display boxes. These groups, or subnetworks of genes, are directly connected to other groups of projected genes by links, which overall provide identification of relaxation and meditation-specific subnetworks. In the vacation-only network on the left, the groups of gene pathways that were deregulated are shown in pink, purple, red, dark blue, and yellow. These are all pathways related to stress and immune processes, including inflammatory response, defense response, and response to stress and wound healing. Overall, the vacation subnetwork shows enrichment for blood coagulation, immune response, beta amyloid metabolism, platelet degranulation, and genes associated with Alzheimer's disease processes. In addition to the baseline changes present in the baseline vacation-only network and the meditation network on the right, additional groups of gene pathways that were downregulated are depicted. Here they're shown in black, blue, salmon, tan, and light green. These pathways were involved in translational initiation, protein targeting to the ER, translational elongation, and viral gene expression in infectious cycles. Overall, the meditation subnetworks show upregulation of genes involved with telomerase ribonucleoprotein enzyme complex, and these genes play an important role in the regulation of telomerase. Finally, I will discuss an example of code used to make this network. I have shown here a simple example, which refers to just connecting 2 independent variables to a single dependent variable for simplicity. So keep in mind that the code to connect many gene subnetworks would look much more complicated. The first line of code from Pomegranate import means that in order to create the Bayesian network, there are packages from Pomegranate that must first be downloaded. Once this is done, the variables need to be created. Here, the first 2 are independent variables, which are set up as previously defined fixed values. The third is a dependent variable, and the value of this variable is set to be equal to the probability table as shown, since its value is dependent on a probability based off of the first 2 independent variables. Next, we have 3 new variables created, S1, S2, and S3. And all 3 of these variables are setting up each of the nodes or boxes that will be displayed in the network and labeling them as independent or dependent variables. The next section, setting up the visual model, is adding code to give the Bayesian network a title, adding the nodes, and creating links between the nodes. The next part of creating the model uses a probability algorithm, so you need to import NumPy and then execute the already created Bayesian network algorithm. The last step of the code is to print the probabilities of each variable's influence over the others, and then use this to make predictions. These predictions are set up where the last piece of data is unspecified, written as none, and the output will fill this in. This leads me to a list of further questions to think about in regards to the Bayesian network used in this study. 1. The example code shown only compares 2 independent variables and 1 dependent variable. How can the code be expanded to include all the variables the study is examining in an efficient way? 2. Is the circular presentation of the Bayesian network the best way to present the data? Or would a more linear shape be easier to follow or better to show how one variable is correlated with another? 3. Could the links be made clearer by using arrows instead of simple lines to show order of effect? And 4. How does the code change when an independent variable affects not one, but multiple dependent variables at the same time? Finally, here are the sources I used to better understand the study and the code used. Thank you for listening, and I'm looking forward to your feedback.